Hi Nat5, so we are still looking at distribution of organisms. So now we need to look at how you can actually sample what organisms are there. So we're going to look at a couple for animals and plants that you need to know, and I've got a few extras that I'm going to show you. Right, so first off, why would you sample in the first place? And a sample is literally just that. It's not the whole thing. You're just going to take a snapshot and then you're going to say, right, okay, if that's what it was there at part of the area, what's it like for the whole of the area? So you might be looking at conditions, so that would be abiotic stuff. You certainly want to know about populations, so that's biotic. You might want to sample stuff about their niche, so exactly how they are fitting into that particular area, like what they're feeding and things like that. And you might sample things like pollution. Now that's probably going to be an abiotic thing that you're looking at in terms of the pollutant, the thing that's actually polluting the area. But it might also be an impact thing. So you might be looking at things called indicator species, which we're going to look at a little bit later on. So the first one that you need to know about is a quadrat. Now I am assuming you remember using these way back in like second year. Okay, so basically it's just a frame of some form that you divide into smaller squares. Okay, so this one's a 5 by 5. Okay, so the potential in this one is to have a maximum abundance of 25 because we have 25 squares. Okay, so you can use it to identify and count basically anything that doesn't move. So generally plants. Okay, but you could use it for things like, you know, like sea anemones that are stuck to rocks. So you should start with putting it down randomly. So generally that's a kind of cast, you throw it and that's it. And then you count how many you have and you get the abundance. But as I say, you only count each square once. So if I had, for example, I was looking for a particular plant and I had a whole pile in here, one in this one, I had a couple over here, right? So in terms of count, this one counts. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And that's it. I don't try and count every individual plant. Okay. So, as I say, it could be anywhere where things don't move particularly well. So it could be underwater. And you take a reasonable number of samples. You've got to make them random. And you've got to be careful about how you count. So generally our rules would normally be you would count if the plant is inside the square or covers half of the inside of a square. Sometimes you don't actually want to be random. Normally we are with quadrats, but you can do something called a transect. And so what that means is that you might, basically, you draw a line, but you use something physically to make the line. And then you take samples along the line. So say you took a line that was 100 meters long and every 10 meters you stopped at that line, you took the temperature, the soil moisture, a whole pile of abiotics, and then you put a quadrat down and you do a quadrat count as well. Or you might just look to see which plants are there, okay? But it could also be underwater again. There's nothing that says the line has to be on land. And you can take all sorts of readings, okay? Our problems with quadrats, well, obviously you need to have something that's about the right size for the thing you want to sample. So it could be very large, it could be very small. You have to have something that doesn't move. Now, you can actually do something that does move as long as you basically take a picture of it and then put a kind of grid on top of it. And they've started using a lot of this from space to do some really interesting stuff about where things are. If you want to avoid the errors in a quadrat, well, it's all the things that we've just said, but not doing anything silly. So make sure you've taken enough samples. Repeat, repeat, repeat. If you are afraid of miscounting or incorrect identification, which is quite common, you need to take extra care. You might need to recount, so you do it through once and then you check it again. If you've got the same answer, you're like, yeah, I think that's my answer. It's really useful to have experts, things who actually say, no, okay, that's not a bluebell, that's a snowdrop, assuming you would know the difference between the two. And so obviously training might be helpful. And if you are trying to do a normal sampling, if it's not random, that's an issue. So you could use a random generator. So a computer basically tells you you're going to go to this point, this point, and this point. Or you can do things like throwing a dice that says 6. You go, I'm going 6 steps, and then you turn around and then throw it again, and it says 2 steps. You do 2 steps. That's you. You know, there's all sorts of ways to make it random. So if your quadrats are for things that don't move, this is for things that move. So what we're looking at generally in your sampling that you're going to be given any information on, it's going to be crawling insect size. So in that case, we're looking at a kind of yogurt pot size, and you might have had a chance of doing this in second year. So what you need to do is make sure that when you dig your hole, you to put your pot into, this actually sits really close to the level of the soil, because otherwise, if it was up here and up high, it would basically block the insect from coming in. It would just kind of crawl around the outside. And you put a lid on top, but the lid is just basically a cover that's camouflaging it and holding it up a bit so they can still get in. So it's not going to be closed off lid. There are some fairly nasty things we do actually. Quite often you'll have something in the bottom here to kill off the insects. Give some thought to why. And the lid is really important, and we'll come back to that in a minute. So our limitations for pitfall traps are, again, I would think kind of obvious. Okay, you might lose animals to other animals feeding on them. Like you could end up basically with one big bug. If you have, effectively you're just making it a buffet. Other animals are just, other insects are crawling in and landing next to it to eat. So that's one of the reasons we might decide to kill. Okay, they could avoid it. So that's why you try and camouflage it and make sure it's nice and neat to the edge. Or they could escape. Now, again, escaping might be flying. So again, another reason why we might go for a kill. And the size of the trap is obviously quite important. So depending on what you want to catch, you need to have a bigger or a smaller trap. I doubt that we're looking at woolly mammoth levels. So to try and avoid it, it's kind of obvious, right? So the same one that comes up every single time. If you've not taken enough samples, it is not going to be representative, so repeat for reliability. So if you don't want to kill them, then your other answer is to go and check them. Check it regularly, make sure that you've got everything out that's got there. Use a lid on the top to make sure that you've got camouflage, because it's not necessarily the one big bug issue. It could be basically you making a bird feeder by producing this. So if you put a lid over the top, then obviously they won't be able to see them. And the animals avoiding, as I say, that's just making sure it's flush with the ground and it blends in. So here are some other methods, not ones that you need to know, but I think worth seeing and kind of, right, okay, so these are other ways we could do stuff. So there is something called a Telgram funnel. And the reason I'm saying that it's worth knowing these is because potentially this could be a problem solving type thing. So what you do here is you put a sample of soil on top of a metal mesh, a gauze, inside a funnel. And then you put a bulb above the soil. And because most things don't particularly like to be dried out inside soil, all the living things in them, they kind of move down the way inside the soil until they get to the gauze, and then they obviously fall through. And then we kill them. Yeah, nobody said biologists were nice. And then you can sample what's there. Putters, you might have seen them in first or second year. So a putter is a pretty simple setup. So you've got a straw basically, which you're soaking through, okay? But you've got a little block on the end of that straw so that you're not going to soak up the insects, because as you kind of soak in air, it means that the air flows all the way down here. So basically you're creating a little kind of vacuum here, so you can point at things and pick them up, and they go into the jar or whatever you're collecting, but not into you, which is good if you're trying not to eat insects. But there is some kind of thought that maybe what we should be doing is considering insects as a nutrition source. And then we've got tree beating, which sounds pretty drastic, but is a very effective way as long as you're careful about you're not doing too much damage to the plant. So you basically put out a sheet and then you hit the tree and things fall out. You have some other versions of this where you might spread the sheet underneath the tree and then you basically let out a gas that will knock out any insects so they just fall out of the tree, and then you collect them. And you definitely might have seen sweep netting, which is what it sounds like. Just sweep through the air and then catch. And it doesn't always have to be in the air. Instead of sweep netting in the air, what you might do is do pond netting. Pond netting is, again, nets, and you basically just pick what size of thing you're trying to catch according to how big the mesh is. And you might have also heard of kick sampling. If you're doing geography, you might have done this as part of your Nat 5 going out and doing your sampling there. Okay, that's us.